Okay, guys, some notes for the cell division unit. A couple of phrases that you're going to want to copy down. Mitosis, which means cell division, two cells splitting, forming or one cell splitting to form two new cells. And meiosis, which is the same thing, mitosis, except for gametes, for sex cells. And it goes through double steps, and we'll get into that, okay? You're going to want to copy down deoxyribonucleic acid, or DNA. This is the stuff that chromosomes are made of, okay? This is your cell's instruction manual, so it tells your cells how to make and when to make proteins. DNA is found inside the nucleus for eukaryotic cells. Okay, when a cell is small, the DNA can meet all of the cell's needs. As a cell grows inside, it does not make more copies of DNA, and so you end up running into what's called an information crisis. Okay, so if you can think of a library that's too big for the number of books, okay, it needs to split into two smaller libraries. All right, chromosome. We're going to want to write this down. A chromosome is DNA in a tightly packed form. Okay, and there's two types of chromosomes. So we have 22 regular chromosomes, and then we have a sex chromosome. Now, that's in gametes. So for somatic cells, we have two sex chromosomes. And then we have 42 regular chromosomes. These floor mats I saw at Pike's Market in Seattle. I'll kind of adjust the screen so maybe you can see that. Okay, that's the male and the female entry into the restrooms. That's controversial these days, so it might not be still that same way. Okay. So cells that are sex chromosomes are called sex chromosomes, and then regular chromosomes are called autosomes. So this is a little karyotype here where they've taken all the chromosomes from a human being's cell, from the nucleus of a cell. They probably did this during metaphase, when they're all lined up right in the middle, and we'll get into that. So each cell has two copies of each chromosome, one from mom and one from dad, as we talked about in our last slide, and those are called homologous pairs. Okay. So you have 44 autosomes and two sex chromosomes. 23 pairs makes 46 chromosomes. That's your diploid number. Now, this is interesting. This is a karyotype of a human being. Now, I can look at this human being and I can see a few things about it. Okay, somebody has used a high-power microscope that's designed, probably a geneticist that's trying to determine for a family the sex of their baby or some other health attribute. But let's look at this for a minute. Okay, you've got homologous pairs of chromosome number 1, and those have millions of genes on them, anything from eye color to how round your elbows might be, to how tall you are, to whether or not your hair is brown or blonde, and how pointy your nose is. So that might all be located on chromosome number 1. And then chromosome 2, there's a homologous pairs. Everything's going great. Homologous pairs 5, 6, all doing great. 7, 8, we get down here to the sex chromosome, and I can see that this is a little girl because it looks like 2 X chromosomes. Okay, if you can see there, 2 X chromosomes. There's one thing wrong here. This is chromosome number 21, and it has 3 copies, which is probably why this karyotype got set aside. Now, when you have 3 copies, you wind up with something called Down syndrome. Okay. So Down syndrome is from having something you go through called non-disjunction, and we'll talk about that. but that's trisomy 21. So anytime you have a trisomy, 3 copies of a chromosome, typically you don't come to term. A baby will be miscarried week 1, week 2, week 3, week 4, whatever. Every now and then, a few of these trisomies are survivable, and trisomy 21 is an example. And you guys all recognize a baby that has Down syndrome. They've got certain traits that are characteristics that are visible. Okay, some things to copy down. So haploid, if you haven't copied it down from the last slideshow, the abbreviation for it is 1N. Okay, your haploid cells are sperm and egg. Diploid, 2N. Okay, so 2 sets of haploids come together to make 1 diploid. Okay, so that's why sperm carries 23 chromosomes and eggs carry 23 chromosomes in humans. We're going to get to this later, but you could write this term down, spindle fiber. This is a little out of order. Spindle fiber is something that's a little bit, I would compare it to fishing line. It's something that you're going to pull a chromosome towards one pole of a cell when it's time for it to split. Spindle fiber. Cell division. When a cell divides into 2 new daughter cells. That's the definition of cell division. And the first kind that we're going to talk about is mitosis. That's when it happens to an autosome. Each daughter cell gets a complete set of DNA, which means you have the appropriate amount of information for that library. And it also keeps a cell from having to get too big to contain DNA. Okay, we use mitosis to replace dead or dying cells, damaged cells. We use it for healing, scabbing over and then healing. We use it to grow, increased cell numbers. Here's the cycles of mitosis. It starts with interphase. Interphase represents 99% of a cell's life. And during interphase, DNA synthesis is taking place. You're getting protein made, all kinds of cool stuff's going on. G2 phase, G1 phase, all part of interphase. Cell division part is very tiny. It's a very small part of a cell's life. And we're going to study that. The final phase is cytokinesis, when a cell splits in half and becomes 2 cells. You can come back to this as it makes more sense. Okay, the one on the left, mitosis, is the first one we're going to study, and that is cell division as it pertains to, say, your scalp or your elbow or any other part of your body. Those are autosomes. Meiosis, we'll study that too, but that's cell division as it pertains to sex. And you can pause this if you need to copy some of this stuff down, which I would recommend. I'm going to continue. Okay, so here's mitosis. Okay, and we'll talk about this in a little bit in a lecture. I'll show you some things you can do with your hands to help you remember this. But prophase is when you, this is just a tiny snippet of a cell's life when it's about to divide. For the first time ever in a cell's life, you begin to see, you can see under a microscope, chromosomes. Normally chromosomes, I would compare them to a slinky that's being stretched out so thin you can't see the wire. And then when you slink it up and bunch it up, you've got chromosomes that become visible, and that's during prophase. Okay? The next phase is metaphase, and that's when the chromosomes all line up in the equator of the cell. And you can see them all line up in the middle there. Anaphase is when the spindle fibers begin to pull the chromosomes apart. And telophase is the last part when it splits. Prophase, metaphase, anaphase, telophase. Okay. This gives you the instructions for how you would describe each one. I would pause this and write these terms down in your notes. I think it would even behoove you to make a sketch. Okay. Cytokinesis is that last part of telophase when a cell splits. And this area right in the center here is what's called a cleavage furrow, that little indentation that's showing somebody that looks at a microscope that it's just about to split. The difference between animal cells that are going through mitosis and plant cells, plants have that cell wall right here. So there's little plates form right here, and that's the emergence of a new cell wall. So that's something that animal cells don't have to deal with because they don't have a cell wall. They just form a cleavage furrow right here, and they split. So here it is shown like you would see it under a microscope. Prophase, the emergence of chromosomes. Metaphase, the lining up in the equator. Anaphase, spindle fibers pulling the chromosomes to the opposite poles. Telophase, it's about to split. Cytokinesis, a successful split, 2 new cells. And then they go back into G1 phase, G2 phase. That's interphase, when you no longer see the chromosomes. Okay, meiosis is just the same as mitosis, except it happens twice. Okay, so you get prophase 1, metaphase 1, anaphase 1, and telophase 1. Then prophase 2, metaphase 2, anaphase 2, then telophase 2. And we'll go into that real quickly. I'm running out of time on this video, so if it seems like I'm hurrying, it's because I intend to make another slide. Okay, so in metaphase 1 and telophase 1, all these things are happening. You'll see that these chromosomes have a blue and a red look. That's to represent the ones that came from mom and the ones that came from dad. Something called crossing over occurs right here. Okay, and this satisfies something called the law of independent assortment, which means chromosomes from mom and chromosomes from dad, they separate randomly. This keeps people or animals from all sharing the exact same genetic traits. As your cells go through meiosis, the chromosomes that came from your mom go to opposite poles sometimes, and the chromosomes that came from your dad go to opposite poles, and they sort of mix match. And that way you don't look exactly like your sister or your brother. Here's an overview of meiosis. Okay. Telophase 1 is when cytokinesis begins, and then it shifts right into meiosis 2. The final product of meiosis is that you wind up with 4 sperm, okay, 4 daughter cells, or in the case of females, 1 egg, and then 3 polar bodies that kind of get absorbed. So you really just wind up with 1 egg. You wind up with half as many chromosomes in meiosis and 4 daughter cells. So meiosis is a little bit different. In mitosis, you wind up with the same number of chromosomes and 2, double. In meiosis, which is for sex cells, you wind up with 4 and half as many chromosomes. I talked earlier about Down syndrome, any of the trisomies actually, and this happens when, for whatever reason, they line up in the middle and then 2 get stuck together. And it's what's called non-disjunction, and you wind up with a trisomy. So this is one of the reasons that Down syndrome is more common in older moms that are having their babies in their mid-30s rather than their mid-20s, is because those chromosomes in their eggs have been together for so long that sometimes they stick, and that's becomes a triple copy. The survivable one being if it's chromosome number 21. Here's little Down's baby. Cute little thing, but you can see the traits, the characteristics in a Down's baby. Okay, we'll do more as we go. Hopefully you guys are learning some of this stuff.